going to be confusing enough forwards. All right, so I'm going to tell you about a new method that I've been developing with members of my laboratory, and I'll show them in the last slide. But I want to just orient you carefully to what the method is, and it's going to involve a little bit of theory, but we'll get through that pretty quickly. So the method is called MAC-PRF, which is standing for Model Average Clustering and Plot-Epsilon Random Field Analysis. And it's a method that we've been developing to detect intragenic selection heterogeneity based on polymorphism and divergence. And before I go on to my slides, I just want to first mention this is very different from many of the things that you've been seeing in the talks recently, because most people aren't working with polymorphism and divergence. So let me lay it all out for you. Recently, that's done a lot less. So if we're looking for ancient selection, then some of the talks you've heard about today are using methods like PAML, estimating omega, or BSRL is another method. These are methods that look over a deep time with a phylogeny to define selection that occurred over much longer time scales. And so what they do is they look, and they actually there are approaches within those that we've heard about a little bit in this session, to do site-wise selection, looking at individual sites and trying to figure out which sites over the entire phylogeny or possibly within regions of the phylogeny are under selection. Furthermore, there are a lot of methods that are sort of all the rage these days to look for selective sweeps. That's on a very recent time scale, so that's what just happened in the last, I'm not going to say how many years, but very recently. And that's also different from what I'm going to talk about, because and you use methods like TGMSD or IHS, population genetic approaches to do that. And they rely on what's happening before selection and seeing a very large number of sites all looking like a sweep is passing through a population or has passed through a population. So in between these are methods developed to use polymorphism and divergence together to try to figure out whether selection is occurring. And the most famous one is the McDonald-Kreitman test. There's a theory that is one way of interpreting the McDonald-Kreitman test called Poisson random field, which I'm going to use for the method that I'm going to tell you about today. So I'm only looking at this, and it's not meant to talk about those other time scales, just to be very clear. So the McDonald-Kreitman test is well established, but has had some people have had various different criticisms of it in various ways. It tests for selection using gene-wide polymorphism and divergence data. So let me just introduce the idea. The idea is you have divergent sites and polymorphic sites. Most of you are probably very familiar with this. And you have replacement sites and synonymous sites. And the point is that under a neutral hypothesis, your ratio of the divergence in non-synonymous sites to synonymous sites should equal the polymorphism you see in non-synonymous sites and synonymous sites. And so it's a very nice setup for testing for selection. You will have then the prediction that a positive selection should yield a situation where the non-synonymous site to the synonymous site ratio is greater in the divergence than the polymorphism ratios, and negative selection would be DN/PS being less than DN/PS in this parameterization. So that's all well and good. The disadvantage with the McDonald-Kreitman test, in my view, is that it assumes intragenetic homogeneity in selection. That is, implicitly, it assumes that all of the sites within a sequence are undergoing the same selection. Now, we know for most proteins that's extremely unrealistic, and what that's going to do generally is weaken the power of the McDonald-Kreitman test enormously, because if you have some sites that are under selection, positive selection, and some under negative selection, your overall number of sites that you get because of synonymous divergence will be not so high compared to the non-synonymous sites. So what we wanted to do was develop a method that would be more powerful than that. And to do that, we're using the Poisson random field theory, which quantitatively estimates genome-wide selection intensity. And what all this really does is it takes those 4 boxes of the Poisson random field and through a number of equations that involve a number of assumptions, which this method is subject to, it estimates, it says what the expectation of synonymous divergence, replacement divergence, synonymous polymorphism, and replacement polymorphism would be. And then these 4 equations have 4 unknowns, so you can solve this very neatly to estimate the level of selection, which is mostly dependent upon these replacement changes that you observe. That level of selection in the Poisson random framework is called, it's the selection intensity, gamma. And here's the formula. I'm sure everyone will enjoy seeing this. This is actually not the original story of our formula. I've done a very little bit of theory, which is to essentially put this ratio right here, which it isn't there in the original method, And that's because we're doing something very special that's different from what Sawyer and Harlow did. What they did was to look at it again gene-wide, just like McDonald and Kreitman, to try to understand what's happening on the overall gene. So Poisson random field framework assumes not only that selection is happening intragenically homogeneous, but every selection event has exactly the same selection coefficient. So we have broken that by saying across the gene, we're going to allow selection to vary. Now, how exactly we do that depends on this. What we've done is come up with a way of assessing the probability of a replacement divergence versus the probability of a replacement polymorphism. Let me tell you how we calculate that probability for one moment, and then we'll get back to that formula. But that ratio is key here. And the point is those probabilities can be attributed for every site and estimate the gamma for each site. It's a gamma that depends on the assumption that there's locality to selection, so nearby areas are under the same selection or similar selection. So to introduce how we do it, and this is the part that mostly I'm just going to ask you to believe, but if you're very quick, maybe you'll get some of the idea of what we're doing. We have a maximum likely clustering method. This is actually published. This one slide is published, so if you want to know more about it, you can look up Zhang and Townsend in 2009. We have a null hypothesis that there's actually no clustering of sites across the divergent sequence. And then we break up the sequence, across the sequence we're looking at, break up any sequence into sort of three parts, a starting part, an end part, and then you can compose a likelihood of an alternate hypothesis to the hypothesis that things are equally distributed, which involves clustering in the middle compared to the two edge regions. And it turns out, in a way that I'm not going to try to explain, that by taking this tripartite division, you can actually explain any clustering as long as you subcluster within each cluster, iteratively forever. You can get any kind of clustering pattern imaginable. And that's really nice. It's very computationally intensive, which is a very big disadvantage, especially for a fairly simple thing to assess. But the nice thing is it's the most powerful way to assess clustering within a discrete linear sequence that exists. So what this ultimately gives you is a probability of seeing something affected across the site. So what this yields is, and then you can actually do model averaging across all the possible clustering models to give you a probability that a site is different. So I'm going to give you some examples of what you get out of this, and that will probably help. So, for instance, model average clustering using maximum likelihood gives a local probability of varying each site. So here's sequence coordinates. Here are variant sites across the sequence coordinates, and this is the probability over this sequence set of sequence coordinates that you see a variant site in that, according to the maximum likelihood ability to cluster it, which means the bigger the sequence you have, the more you can cluster down little parts, but you aren't getting an estimate for every site that isn't locally. It does have an autocorrelation, but you don't have to put it in ahead of time. It's just the most powerful estimate you can get of the cluster. So I can tell you more about that. Anyway, that's all about the published method. The neat thing is, if you take this kind of probabilistic plot of where there's divergence and where there's not divergence, or where there's polymorphism or not polymorphism, you can take those estimated for replacement divergences and polymorphisms, put them into this formula, and get an estimate across sites for selection. Now, this is how the whole approach works. So this is how MAC-PRF estimates local intragenic selection intensity. At the beginning, and I apologize if you can't read this, but you input the sequence alignment for polymorphism and divergence. It extracts synonymous or replacement polymorphism, synonymous or replacement divergence. Each of these is that then we do a MAC-PRF clustering, so we understand across the sequence where do we see clusters of replacement polymorphism, synonymous polymorphism, synonymous divergence, and replacement divergence. Each of those clustering models then are model average. So it's model average clustering. This is actually the model average clustering across the sequence, each of these that you see here. And then you feed those into that PRF model, merge them, and then you can estimate gamma according to the equation based on the replacement polymorphism again and the replacement divergence to yield gamma across sites. So it allows you to sort of estimate what's happening across sites. This gene is zinc finger, and I'm going to talk about it in just a moment. So let me first talk about a Drosophila gene. We had a talk on selection hovensis earlier in this session. This is ACP26AA. It's actually been well known to be under selection. It's one of these sperm lysine protein, very important, understood. It's under selection. And so I just want to show you it yields a statistical significant result with the traditional McDonald-Kreitman test, which means it's under very strong selection. McDonald-Kreitman is a very conservative test. And these are the data that show that. D is 0.001. If you run this on MAC-PRF, you get a very similar result in terms of its intensity of selection, but it's much more local. So these are the four things that you're estimating: synonymous for polymorphism, divergence, replacement polymorphism, replacement divergence. And then when you feed those all into the Poisson random field, you get this profile of selection across sites. And this region in the middle here is probably the region that's under very intense selection. You can see, if you can see well, I'm not sure how well you can see it, there are gray lines here. That's because we can actually estimate confidence as well, because this is a model averaging approach. So we take the top 5% and bottom 5% and say, that's where we don't work. Actually, it's 0.025. But anyway, that gives you a confidence interval. So it gives you a confidence interval as well. Oh, and you can see that actually there is some interesting biology on that. So the interesting biology is just that, well, I'll just say it. Those lines at the bottom were some cut sites, and the selection appears to happen where there's a specific lysine cutting site. So I got to move on to the next thing, which is FCF1, which is the one I showed you at the bottom of that original plot. Is a sulfur regulatory factor, which for many reasons, and if you look in the literature, you'll see this published, people think it should be under selection in Cerevisiae. McDonald-Crichton test does not show it under selection. So I thought this would be a very interesting one to look at. P equals 0.13, maybe indicative, but it's not 0.05, so what can you do with it? Well, if you run MAC-PRF on this gene, what you find is that there's actually very heterogeneous selection across the gene. And this is not actually that unlikely in Cerevisiae, where it has a very high population size, and although people say it rarely goes through meiosis, it has extremely high recombination. So I'm not actually that surprised to see this kind of heterogeneous signal of selection across the gene. So this is a plot I showed you earlier, but these are the replacement divergences. You see, there's the selection. And the last one is right in front of this last little finger. Maybe interest or maybe not. Okay, so I just want to give you one more result, and these are somewhat tentative because we just finished the genome-wide estimation of this, and I was surprised by the result, but in some ways it makes a lot of sense. So if we compare Saccharomyces cerevisiae and Paradoxus, running MAC-PRF on the entire genome, which, because this is very computationally intensive, required a lot of compute cycles, what we get is a picture of how much selection is occurring across the entire genome in the coding sequences. And it's actually remarkable how strong we find selection is under this model. So these are the 4 different categories that we looked at. There's some things that we couldn't estimate, but generally speaking, we found not much negative and a lot of positive selection. Now, this weak positive and weak negative are all in the category that most people would call neutral. Since it's a continuous measure, we can't say neutral, but this is sort of basically what you'd say is neutral changes that are passive sets spreading through the population. And it's important to understand that this is a flux model, which means this is the selection on changes that occur in the population. It's not what's happening to the creature, it's how much is happening, how much changes. And so as much as 43% of coding sequences are subject to positive selection. Again, I chose that word carefully. Subject doesn't mean they're all undergoing positive selection. That means the prediction from Poisson random field theory is that if a mutation passed through and fixed, it would be a positively selected mutation. So that's equivalent to the kind of calculation where people say how many substitutions are selected based on the changes and so about, which is about, often around 50% of substitutions are estimated to be selected as opposed to neutral that change between species. And then you can also look at how many genes have any positive selection or are predicted to have some changes, and as many as 93% of genes are subject to some small amount somewhere of positive selection. So these are some very interesting results indicating that at the very least, when you look intragenically, you should have much more ability to detect population genetic variation. Sorry, you should have much more power to detect selection when you look with an intragenically divergent rather than intragenically homogeneous approach. That's all I have to say, except for to thank the people in my lab who worked on this. Zhang, who's now actually a professor at Beijing Institute of Genomics, Ning Li, who's moved on to another lab but is still up and out a bit with us, and Zi-Ming Zhao in my lab right now, who's working on the last bits of finishing up this project. Yes?
It's possible that I'm tired, but how is this any different than like a standard sliding window analysis?
So it's not really different. I mean, you do sliding window, you get a result. The only issue with sliding windows, and I personally really dislike this, is you have to choose a window size. There is no window size here. This is just the most powerful way to cluster of all ways. So you could do a sliding window and then throw those values of ratios into the Poisson random field equation. You're basically getting the same thing, but this offers like a package where you don't have to impose anything from the start, and it just gives you an answer. I should probably end, but I'd love to take a question. Anyone can leave who wants to. I guess it depends on what your default assumptions about what goes on. So it's not that the negative selection was low that was really surprising. I was actually maybe a little surprised that there was that much positive selection compared to neutral. That is the weak positive, weak negative. And I guess the reason for that is because if you look at all of the papers that have been published in Nature about a couple of years ago, really, where we did the first genome-wide polymorphism versus divergence studies. So they looked at all genes across the entire genome and said, how many are under selection? You get about a 5% rate of genes being under selection for your McDonald-Crichton test. That's because it's a weakly powered test, but the assumption has been that, like for instance, yeast, you couldn't find selection in yeast, and people have talked about that, and that's an issue. When we look at it with this approach, we find a lot of selection. So that's sort of the default that's making me say, that sort of is a surprising amount. I actually don't think it's really a surprising amount, except in contrast to what people have found in previous papers.
So, Poisson random field originally assumed no linkage in independent sites.
Yes.
What's happening here now?
So this still assumes no linkage in independent sites. So really, it's most appropriate under a strong selection weak mutation model where you just have serial sweeps. Now, the issue is that, so if you do this approach, then, and you don't have that situation, it's generally conservative because a lot of the other things that go on with linkage prevent sites from moving through the population as quickly and fixing as quickly. So generally speaking, that means that the weaker estimated selection coefficients actually might be stronger. So it doesn't really change the broad inferences you make. It does have a bias in terms of that gamma as estimated may be a bit low, especially when it's a low selection level. When it's estimated to be high, it's probably not that biased.